Please welcome Philipp Meyer, RWTH Aachen University.
Hello again. My name is Philipp Meyer. I think by now you have heard that I'm head of entrepreneurship at RWTH Innovation. And I think you've also heard that at RWTH Innovation we support 45,000 students and nearly 7,000 researchers in turning their groundbreaking ideas and inventions into startups. And one such startup today is BioThrust. We nominated BioThrust because they are breaking walls in the production of novel biopharma products. BioThrust has developed a next-generation bioreactor with their disruptive membrane stirrer technology, the first worldwide to achieve bubble-free aeration with sufficient gas transfer. And we thought Konstantin should tell you more about that. Thank you very much.
Breaking the wall of inaccessible cell therapies. Konstantin Kurz, BioThrust.
No worries, I'm just here once. So, we as humankind are on the edge of a new era, the era of cell and gene therapies. Breakthroughs in medicine for the first time enable cure of thus far uncurable diseases like diabetes, Parkinson, heart failure, or cancer. It is projected that up to 1 billion patients could finally find relief for a burden that is thus far not solvable. However, so far only a few thousand people have globally been treated with cell therapies. Due to high manufacturing costs, these therapies are locked behind the wall of accessibility. What makes manufacturing cell therapies so complicated? Well, the difference and the unique feature of cell therapies is that the cell itself is the product. So if you want to use these therapies effectively, you need a cell of high quality and high functionality. To achieve these cells, you need great culture conditions. So supply them with oxygen, nutrients, temperature control, and have low shear forces. Current bioreactor systems, as you can see them here on the slide, don't fulfill these purposes. The main issue is that for providing the oxygen, they are using bubbles to introduce the gas and to feed out the CO2, just like you would do at home with the soda stream. This induces high shear forces, which destroys the cells and leads to low processes with low process efficiency. This means that they can only run in small scale. Let's take the example of a type 1 diabetes therapy. If at the current process efficiencies you want to treat just the annual incidence of type 1 diabetes patients, this would mean more than 200,000 ones, each dose costing more than 40,000 just for manufacturing costs. This is a burden that is not sustainable for our healthcare systems. So we thought, why cannot we have an aeration system that avoids bubbles? And that was the birth of the BioThrust MEMSTAIR system. For the first time in the world, we have invented a system that can introduce oxygen to bioreactors completely without bubbles. The system works by using membranes which function as a stirrer in a unique geometry and thereby also mixing the whole system. Based on this unique technology, we have developed the BioThrust ComfiCell system. This bioreactor is a next-generation system that shall become the backbone of future biopharma manufacturing. With our unique technical features, we have set the world record for cell densities. We can completely avoid form formation, which can use up to 50% of bioreactor volume. We can increase gas transfer by 10x, and we minimize shear forces to the minimum level. Let's recall the example of the diabetes therapy. With our technology, we can reduce the number of 1 to 2,000. That is just 1% of current compared to conventional systems. And we can cut costs by at least 7x. With that, we can make these therapies available to patients all around the globe. These breakthroughs are only doable with an excellent team. And the BioThrust team uniquely combines excellence in engineering, bioprocess development, and business development. We are supported by world-leading experts, but even more, we are working with key opinion leaders who are convinced of the need of our system. We've already closed our seed round with Freigeist Capital, and we also were awarded by the EXC Accelerator. The biopharma industry recognizes us as the solution for their therapies. We are working with 4 of the top 20 pharma companies all around the globe, particularly in the United States and Europe. In addition, we are collaborating with leading CDMOs, academians, and the biotechs of the future to develop our system as the backbone of biopharma manufacturing. Our business model is based on the sale of consumables as single-use systems, thus generating recurring revenue. We invite you to become part of our journey. Let's make cell therapies accessible for everyone and break the wall of inaccessible cell therapies. Thanks a lot.
Yes, I'm coming. This is good. I love this when we give access. We have the first question here. Detlef, please, and then Andreas.
Thank you for the presentation. How much is this scalable? What's the largest volume of reactor you're shooting for?
We can pretty much cover everything that's currently also covered by single-use technology, so we can go up to like 1,000 liters at least.
Andreas, please.
Okay, my question ties into that. So you can double the cell density you mentioned. What are the results concerning the pricing then?
We can, depending on the process, we can even go more than double the cell density. There are processes that are currently stuck at 2 liters. We can bring it to 50 or 2,000 liters. So this is like, that's why we achieved this 7x cost saving. So it's a mixture of density and scale up.
Iris?
I'm assuming your customers are the pharmaceutical industry, but is there any specific use case where it's really like a no-brainer for your technology to be adopted?
You mean within the pharmaceutical industry?
Yeah, exactly.
Yeah. Actually, the example of diabetes is the no-brainer because clinically diabetes therapies are working. The question is, how do we bring them to the 100,000 patients out there? And that's why we need our technology. We're actually working with partners in that field to generate the beta cells needed for these therapies.
Luca, you have a comment.
Yes, it's not a question. I think this is also relevant for CO2. You should explore this way to saturate solutions containing CO2 absorbing phases because this could be more efficient than the standard bubbling.
May I comment on this one, on the comment?
Yes, quickly. Yes.
Just quickly, excellent comment, because this is the way why big pharma in the field of antibody manufacturing contacts us. We can make processes much more intensified for antibodies.
Yeah, we have Cyriac and then we will have Stefan. Sorry, Stefan.
How well is the moat here protected around your device in terms of the principle? Are the patents around the principle or about the specific design?
Well, the design of the membrane stirrer and the bioreactor is protected. So we have 5 patent families, and there's also a unique feature how to bring in the gas into like a stirring system without causing any bubbles or issues with the gas lines.
Stefan.
At what stage are you in concretely? What stage are you, and what could go wrong for you before you're really at the market?
I think we have de-risked the technology, and now we're like in commercial scale-up. So we're talking to big pharma. We have actually 4 pilot projects running with big pharma companies, and I think 20 with smaller ones. And now the question is the biggest risk is probably how to get in a market which is obviously more conservative than other industries.
Inga, you are the next one.
Thank you very much. So who is your competitor, or how is the competitive landscape? How do you think about it?
The good thing is there's no company around the world who has a bubble-free system for bioreactor aeration that's scalable. So there's no competitor with the same technology that we have. I think we have the best technology, but obviously you have competitors. So we're competing with Sartorius, Thermo Fisher, Cytiva, and then there's a startup, PBS Biotech, which has a novel stirrer, but no solution for the aeration. And one comment, aeration is needed to scale up beyond 2 to 10 liters.
Do we have a last question for the last 50 seconds? Anyone? Even the audience? Yes, the audience, please, 3rd row here. No, you need a microphone. Even if you are only the audience, we are all experts.
Thank you. How strong is your IP on that?
So we have 5 patent families. The most important one is the stirrer that is already granted in the United States, South Korea, and the rest will follow. So I think we're pretty straightforward on this.
Well, you're so fast. Okay, we still have 25 seconds. Any other question or comment? Yes, wonderful. This block is very silent. So let's.
What's the life cycle on this thing?
This is a single-use product. This is baseline for pharma industry.
Right.
Fantastic. We have 10 seconds left. Can you make it faster? I think no, we're done. 5, 4, 3, 2, 1, 0. Thank you so much.